So thank you everyone to be here today. This is the first webinar of the new ANDO webinar series on innovation and therapeutics with the pharmaceutical and biotechnology companies, clinicians and researchers. So for this first webinar, I'm very pleased to introduce you to Michael Bauer, MD, PhD. Michael is Vice President of Clinical Development and Medical Affairs of TYRA Biosciences. Michael is a pediatric geneticist, a former director of skeletal dysplasia program in Nemours, and world-known expert on skeletal dysplasia. Today, Michael will introduce us to TYRA Biosciences, the company research focus, and will take us to know more about TYRA 300 development. Michael, welcome. The floor is yours.
Thank you, Inez. We very much appreciate the opportunity to talk to you about what we're doing. I hope you can see my screen now.
Perfect.
Perfect. All right. So let's begin. So what I'd like to tell everyone about today is a little bit of the physiology of endochondral bone formation, specifically how the growth plate works, why FGF signaling in the growth plate is important, and why tyrosine kinase inhibition makes sense for modifying growth plate function, specifically in achondroplasia, and then move on to talk about our specific molecule, TYRA 300, which is an oral tyrosine kinase inhibitor that has specific selectivity for FGFR3. Be able to show you guys some of our preclinical work, and then we can have a discussion based on all this information and whatever questions you may have. So the crux of the matter is endochondral bone formation. This is a critical biologic process. It is very important in many different types of growth, and it all comes down to this organ that we can see under the microscope and that we're going to talk a little bit about over the next 10 minutes or so. And that these are cartilage cells in a growth plate. They're organized into different zones: a resting zone, a proliferating zone, and a hypertrophic zone. And endochondral bone formation is important in the base of the skull, where it helps to shape the size of the foramen magnum. It's important in the midface, which is where all of our nasal passages and breathing passages are. It's important in the spine, both for the height of the vertebral bodies in the front part of the spine, but in the back part of the spine, endochondral bone formation plays a large role in the way the canal is structured. There's an important role in the pelvis and the pelvic outlet, and of course in the extremities, which can lead to shortening. But all of these come back to the same important biologic process. So these are some cartoons that I want to use to illustrate this principle. So this white line here is the growth plate. And what you see is as this bone gets longer, the green is new bone that the growth plate makes. Here, on lower down, there's a different way that bone gets added. And then you can see that the bone itself becomes sculpted so that as it elongates and gets wider, the shape of the bone stays the same. And here is a cartoon of what we just saw in that previous slide, where there are proliferating chondrocytes, hypertrophy chondrocytes, and then the mineralizing cartilage. This right here is our white stripe. And then this gives you an idea about what actually is going on. So these resting chondrocytes begin to divide and they line up, they proliferate, they hypertrophy and they turn this blue color. And the hypertrophy part of the cell is actually the matrix that makes up the bone. It is the bone length. And you can see how the bone gets laid down and gradually mineralizes until the growth plate disappears, which occurs at puberty. So this is what's going on, not just in the long bones, but all of the regions that I just mentioned prior: skull base, spine, pelvis, extremities. This is the process by which we all grow. If you look a little bit more carefully at the spine and the vertebral bodies, we see that the spine increases its height. This is our sitting height, right? And then there's also this red line here. This is a growth plate by which this pedicle gets longer, and that controls the size of the spinal canal. As we know, this process is different in achondroplasia. We'll discuss why in a moment, but it often leads to problems that require surgical interventions. So here's our cartoon again, our bigger version of this. And as you see these proliferative these cells proliferate in the proliferating zone and then become hypertrophied cells, you can sort of imagine that there's a rate. The faster these cells divide, the longer the bone can become. And what we know about this process is that there are two competing signaling pathways. One of them is the FGF or FGFR signaling pathway. And that pathway and the activity, the signaling through that pathway, controls the rate at which these cells turn over, and therefore the rate at which endochondral bone formation proceeds. So if we look at the biology of people that have FGFR3 skeletal dysplasia, this would be the typical pathway that we see. So there's an FGF ligand that binds to an FGFR3 receptor in one of these chondrocytes. It leads to these signaling pathways where it causes the cell to actually stop proliferating or stop hypertrophying. That is, this signaling pathway slows the rate of growth. If you have a mutation in FGFR3, these mutations lead to overactivation of these pathways, and hence we have a break on the rate of cell division, and we have a break that's inappropriately applied to endochondral bone formation. So we asked the biologic question here, with all of these FGFR3 conditions that share the same physiology, is in fact there a way that we can turn off this break and restore the rate of endochondral bone formation? Well, there are drugs that you can use to inhibit FGFR3 signaling. But FGFR3 signaling, or I should say FGFR3, belongs to a family of receptors that are very similar. And if you inhibit one, you usually inhibit more than one. So if you inhibit FGFR3, you can also at the same time inhibit FGFR1, which can lead to an increase in blood phosphate levels. If you inhibit FGFR2, it can lead to dry mouth, dry eyes, dry skin, different types of fingernail issues, toenail issues. And if you inhibit FGFR4, it can lead to GI problems like diarrhea or liver toxicity. So what we set out to do, for many reasons, not the least of which turns out to be that FGFR3 is very involved in cancers, different types of cancers, is we set out to develop a molecule that was selective for FGFR3. That is, we wanted to see if we could develop a compound that would turn off FGFR3 while at the same time not turning off FGFR1, 2, and 4. And the medication or compound that we developed, TYRA 300, is oral. And here you can see that when we turn off FGFR3, we need a significant higher concentration in order to affect FGFR1, 2, and 4, right? So when we have enough drug concentration, this is done in vitro, in a dish, in a cell model. When we turn off FGFR3, we need 19 times as high a concentration to turn off FGFR2. We need 63 times that concentration to turn off FGFR1 and 55 to turn off FGFR4. So we're very selective. And you can compare that to the same experiments that used infigratinib. So here you can see for infigratinib, you need a fraction. So it is impossible to turn off FGFR3 with infigratinib and not also inhibit FGFR2 at the same time. We're only twofold away from inhibiting FGFR1. So we think we have this molecule that is very selective for our targeted receptor. If we give this molecule to mice, and this was done in your part of the world, this was done in Paris, in Dr. Leger-Malay's lab. Here you can see the essentially achondroplasia mouse on the left, and here you can see the achondroplasia mouse, which was given TYRA 300. And you can see that the mice are significantly longer, about 18%. And it's worth saying, so we used 1.2 milligrams per kilo per day for 15 days. So these mice got treated from day 1 to day 15. Not only did we see overall improvements in the lengths of the bones and the spine, we saw significant improvements in the foramen magnum in these mice. So here you can see the foramen magnum from front to back. Sagittally, we have really normalization. And when we look transversely from ear to ear, we see improvement, but not quite back to the typical levels or typical sizes, but definite improvement. So all of this taken together has given us good reason to believe that we're on the right path. It's worth saying none of these mice had any issues that were identifiable in terms of toxicity. So our goal is to move forward with a clinical trial to test TYRA 300 in individuals that have achondroplasia. And what our strategy is to look at more than just average growth velocity. Sure, we like people to be taller. That's okay. But we're really more concerned with how people function in the world. We would like this treatment or potential treatment to minimize the need for daily adaptations. We would like to eliminate the need for reach tools, differences in that are related to stride length and gait. whether that could be fatigue, whether that could be the need for a lower step riser, but many different elements or aspects of function. And we'd also like to see a difference in ultimately pain, because we believe, or we want to see, we definitely want to study the spine and hope that TYRA 300 can affect the way the spine grows. So there was a question that Inez had about what other potential disorders TYRA 300 could treat. Well, we think any FGFR3 associated condition where there is that overactivity in the pathway where we can turn the pathway off has a potential. So that would include hypochondroplasia, thanatophoric dysplasia, Sedan syndrome, as well as potentially double dominant achondroplasia. So all of these conditions have potential. There are also several FGFR3 related craniosynostosis syndromes, including Crouzon with acanthosis nigricans and Munke syndrome. These are all potential conditions where we would directly intervene in this pathway that is different because of the genetic mutations. Now, this pathway, however, exists in every single person's growth plates. So there are other genetic causes of short stature where intervening and modifying the rate of cell division may be useful. And these could include things like Léry-Weill dyschondrosteosis, Turner, osteogenesis imperfecta, different types of the MPSs where we have specific involvement in the skeleton, for example, Morquio, potentially Laurent syndrome, which is a growth hormone insensitivity syndrome. And then even more broadly, perhaps this could play a role in idiopathic short stature down the line. So clearly our first focus is on achondroplasia, but there are other potentials, on this panel, and we'll know a lot more as we start to begin the process and learn more and more about safety profiles and precise dosing. So that's the presentation that I've prepared. We have some people from TYRA on the call, and between them and myself, we are happy to take any questions and have some discussion. I'm going to stop sharing and we can do that.
Thank you so much, Mike. It was really very clear and really visual presentation. I have to say that many years after seeing the explanations of bone growth, this was one of the most easy ways to see how things work. So really appreciate it. It was in our webinar. And thank you also for tackling some questions. In this webinar, I did collect questions from ANDO members, and as to facilitate this Q&A to be more one-to-one. But if there is any question from the audience, I'm sure that it's okay for you to also answer. So firstly, again, thank you for the presentation. It was really good. So my first question goes towards what were the factors that TYRA took or criteria for TYRA to work on FGFR mutations?
Yeah, so the FGFR3 signaling is really critical in human biology. And TYRA, the company, really began its journey into the biosciences, if you will, as an oncology company. And it turns out that overactive FGFR3 mutations are an important driver of many types of human cancers. So the truth is TYRA 300 was initially developed, and other molecules that TYRA is developing really were developed with an eye on oncology, hepatocellular carcinoma, different types of urothelial carcinomas can all be driven by mutations in FGFR3. So it wasn't long after that process began when people realized that this drug not only could potentially be used in cancer, and for that matter, it is in clinical trials for cancer, but also be used for a different kind of targeted FGFR3 overactive biology, which turns out to be really helpful for us and in the skeletal dysplasia population that we are all part of.
Yeah, for sure. But then the question, I mostly think that it's easier to tackle skeletal dysplasia with single point mutations. Not saying that it's easy to work on. It's easier maybe comparing to other genetic mutations. But the challenge here for TYRA's decision to cross for achondroplasia, gladly, it was gladly for the decision, it's a challenging period because there are many other companies working on this specific skeletal dysplasia, achondroplasia. So how confident is the company on the potential outcomes comparing to the other companies working on the same arena?
Well, I think it's fair to say that we wouldn't be doing this if we weren't optimistic about what TYRA 300 could bring. Of course, everything is speculative, but what we think we have that differentiates us from others is precision. Is this molecule is acting as close to the biologic difference as is possible. If you think about the CMP analogs, so you think about vosoritide and the Ascendis drug, they don't work on the FGF braking pathway. Right? Yeah. They work on a different pathway in the growth plate. Now, there's some interrelationships, sure, but they work by a very different mechanism. TYRA 300 goes right to where the mutation, right to where the biologic difference is. And unlike the QED drug, which is non-selective, it turns off FGFR 1, 2, and 3 very close to equipotently. We think that we have a molecule that's just going to get to the place and it's not going to inhibit other receptors. And we think that is going to create, that's the biologic advantage of TYRA 300. I think the other thing that we hope will be able to differentiate us from the other drugs going forward is we are very focused on improving function. Mm-hmm. We want to see that TYRA 300 improves people's lives. And that may or may not be equivalent to being taller, right? It doesn't have to be. Perhaps somebody is more proportionate, but not significantly taller. There's a lot of ways to think about it. I think, the spine is a great example. If we can impact the spine, if you don't end up being taller, but you need less back surgery, that would be a significant improvement. It's really unlikely that any of these things are going to occur in isolation, but we really want to make sure that at the end of the day, we're not creating people that continue to have disproportion that are a little bit taller. We want them to be healthier and more functional.
That's a very important point, and it's coming to a more wider knowledge that it's really important functionality rather than just thinking on longitudinal height, and not stopping the research on just growing inches or centimeters. That's a very important point. One of the things that you presented in that I really like to see was that one-to-one comparison between the triggering of FGFR 1, 2, and 4 on a TYRA 300 and infigratinib. And I wonder, because the research team was is basically the same, is the mouse model the same, or the one used for infigratinib was a mouse model reproducing thanatophoric dysplasia, not achondroplasia, so effects could be ampered? Is this mouse?
So that's it's a question I can speak to a little bit. So the highest level is almost a joke, but it's that mice aren't people.
That's a very important point.
And it all comes back to that. So the idea is that we as scientists, in order to create a mouse model of human skeletal dysplasia, need to activate FGFR3 in an animal, in a mouse. And people have tried for literally decades to put different mutations into mice and see what happens. Well, if you put the exact achondroplasia mutation that humans have into mice, it turns out mice aren't tremendously different. So the mouse model that is used by Dr. Leger-Mallet in the Imagine Institute actually does have a thanatophoric. If that mutation was in a person, they would have thanatophoric dysplasia. When that person gets put into a mouse, or I'm sorry, the person doesn't get put into a mouse. Okay. When that mutation gets put into a mouse model, it looks like achondroplasia. And that's just a difference in the biology, and gets back to, mice aren't people.
Okay.
So that being said, when Dr. Leger-Mallet does these types of experiments, it is the same mouse model. It is what we call a knock-in model, where they add a mutation in, and that mutation is a human thanatophoric mutation, but that really equals achondroplasia in a mouse.
Clear.
And then so that's what your model is. She's been using that model for a long time, and it's pretty well described. And then, you give the mouse some sort of intervention, and you see what happens. So that mouse model was used to study vosoritide, and that data is published. That mouse model was used to study infigratinib, and that data is published. That mouse model was used to study TYRA 300, and that data has been presented at conferences and will be published in the near future. It was, I think, presented for the first time in October at the ASBMR.
Okay.
So, in that data, at least part of it are the slides that I showed you.
Thank you very much, because it's not that easy access to have this initial information on publications. I know that takes a lot of time, so it's good to know that it's already being shared in some events and now here. So it's really good. Taking for another point on your presentation that I really like to see was on the foramen magnum measurements. And in fact, that's a very critical point in people that are born with achondroplasia. It's those critical first years with foramen magnum stenosis. So very glad to see that TYRA is looking closely to that. Has or is any planning to do in your clinical trial that you are planning to also evaluate spinal stenosis or measurements in lumbar spine of the children that will be involved, or just the foramen magnum area, depending on ages?
Yeah, I think the depending on ages is really important. So forget about intervention right now for a second. It's really important to understand sort of the natural history of what happens. So as you mentioned, foramen magnum stenosis and its sequelae are really important and really life-threatening problems that exist in really infants and very young babies with achondroplasia. It's a problem primarily of the first 2 years. Certainly, you could say by the time a child gets to be 3, it's highly unlikely that they're going to have a foramen magnum problem if they haven't already had one. The spine is a little bit different because spine growth, the spinal canal itself grows all the way through teenage years. In terms of the bony canal, there's other differences that exist in the intervertebral discs and some of the ligaments that are inside of the canal. So whenever we make a trial design, it's very important to match the age of the trial participants. So if we were to do a trial, let's say in a 20-year-old, looking at the foramen magnum would be pretty useless, right? That problem was 17 years ago, right? So I think we're aware of these as being, significant problems. The biggest reason that children with achondroplasia die is foramen magnum stenosis. One of the biggest reasons that people have significant pain and limited mobility is because of spinal stenosis. We know these as facts. So it's up to us to figure out how we can study these things when we make an intervention. And that's based in no small part on the age of the participants in the trial. So I can tell you that no, we're not going to be studying foramen magnum stenosis in anybody over 3. It's not a helpful study procedure. When we do get to under 3, undoubtedly that will be in strong consideration to be an endpoint. And conversely, as I mentioned, we intend to study the spine. We intend to hopefully be able to demonstrate that TYRA 300, if given at the correct dosing, can improve the spinal canal in such a way that we minimize spinal stenosis.
I think that would be a brilliant outcome for a research plan. It will take a long time. We expect this to be a very longitudinal, long trial, but I think it's essential, as you mentioned very well. Natural history is critical, and there's still a lot to know for achondroplasia, although comparing to mostly all the other skeletal dysplasia, chondroplasia is one of the best known, but even so, a lot to understand. And now regarding to practical levels, TYRA, with all this progressing, positively progressing research on this molecule, when are plans set to start a clinical trial in your positive predictions?
Well, this is we talked a little bit before about there may be some questions that I have a limited ability to answer, and all that we can really say at this point in time is that we hope in the second half of 2024 to file our IND with the FDA, and that would allow us to begin trials in the United States, and then hopefully soon thereafter in other parts of the world.
Are you considering, and I'm sure yes, regarding who will the children that will be adjusted for this precise clinical trial that you are now preparing? Is it possible to know if children with previous limb lengthening or previous other molecules during clinical trials or not, as one drug is already available, are these children eligible, if you can answer, or they are not?
So ultimately, the clinical trial is going to have to be, let's call it certified either by the FDA or the EMA or both. We hope to be able to include children that have had prior exposure, especially if that intervention did not seem to help them. But that is not entirely in our hands.
Yes.
In terms of limb lengthening, I don't have a clear answer at this point.
Okay, fair. Which are the challenges that you are expecting that TYRA will face in the next months regarding not the submission for FDA and the EMA, but for the clinical trial itself, because it's the ultimate goal is to have an effective clinical trial. What are the challenges that you are predicting?
Oh.
I asked tricky question. Tricky, no. It's a really important question. So,
well, I think one of the concerns is that there are, as you know, a lot of other drugs, or maybe not a lot, but several choices. Yeah. So people could already be in a trial. People could choose to not be in a trial and have their children on something that has approval. So enrollment could potentially be a challenge. The number of centers that we think are going to be able to actually perform at a high level a clinical trial. This cannot be done at every local doctor's office. So all of these factors, I think, make it challenging, but I do think we can do it.
You always have to have a very positive, as you said, very positive expectations. Otherwise, you wouldn't be there. So that's a good point. And regarding other skeletal dysplasia. You presented a very clear chart and table with several other skeletal dysplasia beside achondroplasia. Knowing that going to a clinical trial, you need to show proof of concept and results to the medicines agencies, and that takes a lot of time. How expectable is for TYRA to take other dysplasias, for instance, hypochondroplasia or Munke or Larry-Weil chondrodysostoses, many others in the future years? How can this work out for TYRA?
That is a real crystal ball question. If you have the I don't know, is there a lottery in Portugal?
Yeah, and Portugal and that European, so national lotteries and European lotteries. So
yeah, so if you can tell me the lottery numbers, I might be able to tell you what's going to happen in the future there. You know, I think one thing leads to another, though. If the more success we have, the smoother things go with achondroplasia trial, with safety data, with dose finding, the more likely it is that those other things can proceed. But, you know, we have to do one thing at a time, and we're clearly focused on achondroplasia to begin with. And like you know, all these other things exist in our thinking, right?
That's a good starting point, because if the company didn't have any overview on other possibilities on other skeletal dysplasia that could be tackled with the same molecule, we would have a different conversation. But it's really good to see there is unmet need for the majority of skeletal dysplasia, and knowing that a molecule as TYRA 300 has potential to tackle FGFRs, specific skeletal dysplasia, it's that possibility that is in the scope on the company's positive, and we'll have to also, I'm aware, wait for these results, but nevertheless asking questions. I have parents with children with hypochondroplasia also requesting this information, and yeah, it's something that it's natural.
It's a very good question. It's a natural, you know, progression. It's a good question. We just need to go one step at a time.
Do you think it will be more challenging if TYRA tackles a skeletal dysplasia in this mutation FGFR3 that has a limited number of participants, like a very reduced prevalence compared to achondroplasia, or that won't be an added challenge?
No, it clearly would be an added challenge. I think it's such a difficult question to answer, because you know, when you sit in my chair, you have to start. I know from my days in the clinic, I understand hypochondroplasia, I understand thanatophoric dysplasia, I understand double dominant. But when you sit in the chair of trying to develop a molecule and develop a clinical trial, you really have to think about, how can I demonstrate that this molecule that I'm giving somebody makes them better? And we'll just leave better out there as to what that means. The less children that you have to study, the harder it is potentially to show that there's a difference. And that's, you know, that has nothing to do with TYRA, and it has nothing to do, that's just the reality of rare disease. And it's something that we struggle with. It's something that the regulatory agencies have tried to help with trial designs that are a little different. And, even achondroplasia, where there are literally tens of thousands of children around the world, if you think about it, you think about typical health problems, high cholesterol, diabetes, blood pressure, Alzheimer's. There are literally millions of people in the world that have these problems. And when people do clinical trials, they do trials with 10s and 20s and 30,000 people in some of these trials, right? We don't have, it's such a different world when you get into rare disease. It's just a different landscape, and that's the world we live in right now, and we have to figure out how to navigate that, all of us, as parents, as drug companies, as clinicians. How do we figure out what's the best thing for our children, for our patients? It's challenging, but it's fun.
Definitely. And another difficult question. Do you think that, and again, looking to your crystal ball, having different skeletal dysplasia in that list that actually exists with lesser individuals that have those dysplasia, having, instead of a multicentric clinical trial, having a multi-condition clinical trial with minimal numbers, because you're using the same molecule and also working on different conditions, different diseases. I'll do envision that possibility. It's more practical. I'm thinking on a practical level, without being harmful. If the first clinical trial with achondroplasia show no harm, no side effects, how potential could it exist in the future, a translation to also faster development, tackling different skeletal dysplasia in the same clinical trial? Do you think that's a possibility, or is totally unlikely?
Boy, that is a real crystal ball question. I don't know the answer. I think, all of us, again, these different stakeholders, parents and support groups and drug companies and clinicians, I mean, in some ways we all share the same goals. Like we want our children, our patients to have better lives. And we are not free to operate in a world without controls, right? So there's the EMA has controls about how you do trials, and the FDA in the United States has controls about, how you do trials, and we have to work in those parameters. Can we be innovative? Can we be clever? Can we, do things that are maybe a little unorthodox? Yes, with their permission.
Exactly. Exactly.
But we can't just go around and, make stuff up.
No.
So you have to have a real good rationale for why your potential drug is going to work. How are you going to measure it? And if there's a way to group some kids together, then to increase numbers or things like that, then you can do it if it makes biologic sense and you have these approvals.
Okay. And now, no crystal ball. You showed a last slide with possible outcomes or essential aspects that TYRA wants to focus on this research. It's longitudinal growth, reaching objects, functionality. I really like to see stride and pain as a focus on also on the outcomes of this research. When you mention reaching, are any expectations on proportionality, or have you seen any data in the mouse model that leads towards that?
I would, so the second question first is, I would go back to my statement that mice aren't people.
Yeah.
And I don't, the mice have more proportionate short stature than humans do. So I don't know that we can, I think that's a little apples to oranges. But yes, to answer the first part of the question is how reach. So there's in the endpoint world, in the clinical trial world, there are measurements. There's anthropometric measurements. How long is your humerus? How long is your ulna? How wide is your arm span? These are measurements. These are numerical. In a functional world, you have reach. So reach is a measure of function. Lengths are a measure of growth. So when we talk about, and you talk about proportion versus disproportion, to some extent, baked into that is the idea that we're going to compare one measure to another. That's not actually what reach is. Reach is more about, for example, can you pull the quarter out of your pocket? I don't know what to change. Can you pull the euro out of your pocket?
Yes.
That is reach. How far can you move your hand from your shoulder? That is reach. It's different than the measurement of the different bones. So proportionality is really baked into the concept of reach, if that helps.
Yeah, because it's a factor that is usually untackled, and it's very, very important for functionality. But how hard will it be to measure functionality?
Ask me in 2 years.
Okay, Mike, this has been brilliant. I just wanted to say, because you're expert in skeletal dysplasia, but also an experienced pediatrician on genetic pediatrics, what are your considerations on this state of the art currently, from all the years in clinical practice until 2024? What are your real expectations for children that are being born now with achondroplasia?
Well, I started in this business probably about 20, 25 years ago. And at that point in time, care was entirely symptomatic. We had very limited natural history data, even for achondroplasia. And if we move as you know, my career went forward and time went forward, we developed natural history data. And then, you know, in the last, whatever, 5 to 10 years, all of a sudden we understood molecular biology and we understood the pathways, and we saw people, companies developing various drugs that could modify these pathways. And now all of a sudden we're literally living in an age where there's a paradigm shift, and we are shifting from entirely symptomatic care to care that I'm not sure that we can quite call it prevention care, but definitely, care where we have the opportunity for some medications or potential medications to change the natural history. We now are living on the edge where we can change natural history, and I think that's exciting. It's scary, and it's exciting. It is a new world.
For sure.
So, yeah.
I'm glad that you think that this exciting. I'm really glad to see also all this development from 11 years ago when I was confronted by a diagnosis of achondroplasia to one of my kids, and seeing all this developments, and also a thank you note to all the researchers, because all the research around achondroplasia made, as you said, change natural history, because now we have multidisciplinary teams that did not exist years ago. Also, always caring for the better development of children. So now, thank you very much. This was a really, really first great webinar. Thank you to TYRA. Thank you so much, Mike, for being the speaker and all this unpredictable questions on the future. Let's see how future works well for TYRA 300, and good expectations here. And thank you again.
Thank you. Good to see you.
Bye.
Bye-bye, everyone.